Today we are going to talk about the fascinating roles of enzymes in the process of fasting. Our video is entitled as Enzymes Involved in Fasting. When you hear the word fasting, what's the first thing that comes to mind? Is it a religious practice, a cultural tradition, or something purely scientific? The answer is it's both. Fasting has been practiced for thousands of years for spiritual and cultural reasons. But in recent decades, scientific research has revealed the incredible psychological and metabolic effects of fasting on the human body. Whether we see it as a ritual or science, it's a powerful practice with measurable effects. Before we dive deep into the specifics, let's understand the basic role of enzymes. Enzymes are biological catalysts that speed up the chemical reactions in our body. In the context of fasting, they are crucial for regulating our body's metabolism. During a fasting, our body shifts from using glucose as its primary fuel source to using stored energy. Enzymes are the key players that manage this transition, ensuring we have a continuous supply of energy even without eating. Fasting triggers the release of hormones like glucagon, which signal our body to tap into its energy reserves. These hormones work directly with enzymes to facilitate processes like gluconeogenesis, lipolysis, which we will discuss in more detail. Enzymes like lipases and amylases become active to break down stored fats, glycogen, making sure we efficiently use our internal energy stores. Fasting is not just about restricting food, it's about activating key processes in our body that leads to significant benefits. Fasting enhances mitochondrial function, improving our cells produce energy. By promoting the breakdown of fats, fasting helps in fat oxidation, can reduce overall body fat percentage. It can increase our metabolic rate, leading to more efficient nutrient use. Fasting optimizes brain function and cognitive performances, which is why many people report feeling more mentally clear during fasting. The process triggers autophagy, a cellular cleanup process that helps remove damaged cells and can contribute to a longer, healthier life. Now, let's look at some of the specific enzymes that are critical during fasting. First one is glycogen. Although it's not an enzyme, glycogen is the stored form of glucose. Enzymes like glycogen phosphorylase break down glycogen to release glucose for energy. Next one is AMPK. This is the master regulator of energy balance. AMP activated protein kinase helps the body switch to burning fat when glucose is low. Lipase are a family of enzymes that break down stored fats into fatty acids, which our body can use for fuel. The last one is glucagon, another key hormone that signals the liver to release glucose. There are other sets of enzymes too. First one is CPT1. This enzyme is essential for transporting fatty acids into the mitochondria so they can be broken down for energy. pKa. This enzyme promotes the creation of new glucose from non-carbohydrate sources, a process called gluconeogenesis. Third one is PEPCK, a critical enzyme in the gluconeogenesis pathway. The last one is LDH. This enzyme helps in the anaerobic metabolism of glucose, providing quick energy. Now we will see the role of gluconeogenesis in fasting. When we don't eat, a major challenge is maintaining stable blood glucose levels. Without this, our brain, which relies on glucose, cannot function properly. This is where gluconeogenesis comes in. Gluconeogenesis is the process of creating new glucose from non-carbohydrate sources like amino acids and glycerol. This process is a solution to the problem of low blood glucose, and it's essential for maintaining stable energy levels during fasting. It's a lifesaver for our brain and other glucose dependent tissues. Now we will deep dive into the metabolism pathway of glycogen, which is called glycogenolysis. The first line of defense during fasting is breaking down glycogen, our stored glucose. This process is called glycogenolysis. During fasting, our body is triggered. When blood glucose levels drop, our body releases glucagon and epinephrine. Glucagon binds to the receptors in the liver, activating a series of enzymes, which leads to the stored glycogen is broken down into free glucose, which is then released into the bloodstream to raise blood sugar. Once glycogen stores are depleted, our body turns into fat. This is where hormone-sensitive lipase, HSL, becomes a hero. Now the next step is activation. Hormones like glucagon, epinephrine and cortisol activates HSL. Next, the HSL breaks down the stored triglycerides into fatty acid and glycerol, here which is shown in detail. Now, the fate of glycerol is explained in detail, where we can see that glycerol can be converted into glucose in the liver, contributing to gluconeogenesis. And the fate of fatty acids is fatty acids are transported into the mitochondria and used to produce a large amount of energy through a process called beta oxidation. Some are also converted into ketone bodies in the liver, which can be used by the brain for fuel. In prolonged fasting, our body will start breaking down protein into amino acids, a process called proteolysis. Now we will see how this happens. The first step is triggering. This happens when both glycogen and fat stores are running low, and the purpose is the amino acids are used for gluconeogenesis, ensuring the brain still has a supply of glucose. Amino acids are classified as glucogenic, ketogenic, or both, depending on how they are metabolized. This ensures our body can continue to produce energy and essential compounds. To summarize, when we fast, our body activates a series of metabolic pathways to maintain energy balance. The pathways are mentioned as below. Glycogenolysis, which is breaking down glycogen for glucose. The next one is lipolysis, breaking down fats for fatty acids and glycerol. Proteolysis, breaking down proteins for amino acids. Gluconeogenesis, creating new glucose from non-carbohydrate sources. Ketogenesis, converting fatty acids into ketone bodies for brain fuel. And the last one is oxidation, using fatty acids and ketones for energy. Now this is one of the most exciting aspects of fasting, autophagy or self-eating, which includes 4 steps. First one is initiation, where fasting triggers a cellular stress response, signaling the need for a deep cleanup. Next one is activation. Enzymes like AMPK are activated, which initiates the process. Third step is degradation. Autophagy enzymes break down damaged cellular components, organelles, and misfolded proteins. The last step is recycling, where the resulting amino acids and fatty acids are then recycled and used for energy production and to build new healthy cells. This is a crucial process for maintaining cellular health and promoting longevity. In conclusion, fasting is a temporary restriction of food and drink that triggers a remarkable hormonal shift in our body. This leads to a series of metabolic adaptations, allowing us to use stored energy efficiently. The health outcomes are significant, including improved insulin sensitivity, reduced body fat, and better overall metabolic health. Thank you all for your time and patience and watching this video.